Welcome to Witham Sounding Board, a podcast sharing powerful business tips, insights, and trends for those seeking to become a rock star in their industry.
Hello and welcome back to Witham C-Suite Podcast. I am your host, Cody Thomas. I am part of Witham's outsourced accounting and fractional CFO service line. We support the offices, controllers, and CFOs. If you'd like to know more about what we do and how we support our client, please go to witham.com/oasis. And that's spelled OASYS. Joining me today is Dom Hebrold. He's an Associate Director of Process Technology at Veronova. Veronova is a US-based CDMO specializing in emerging modalities such as CIDES, ADC, and PROTACs. Don has an extensive background in digital and laboratory technologies in biotech and pharma, and has held leadership roles at CDMO, which is Veronova, where he's currently at. Large pharma like Boehringer Ingelheim, and technology partners, Metla Toledo. He's passionate about leveraging advanced tools such as AI, automation, and data analytics to optimize drug development. He has extensive experience in conducting drug R&D in space in microgravity. My discussion with Dom today will explore his journey, which is pretty good. The science behind biotech innovation and the technologies shaping pharmaceutical manufacturing. Dom, my friend, welcome. How are you?
Yeah, thank you very much for the kind introduction, Cody, and thank you very much for Witham for hosting me on this podcast. It's a true privilege to be able to share my passion and my motivation for life sciences and biotech in particular. Yeah, so I'm currently indeed Associate Director of Process Technologies at Veronova. So I'm going to define a few words. Process technology means anything beyond the test tubes, all the complex technologies that the biologists and the chemists will not spontaneously use because there is a activation barrier you have to learn. There is some data science behind that. I'll come back to that. Okay. And Veronova is a US-based CDMO. CDMO stands for Contract Development and Manufacturing Organization. And so we are Massachusetts-based, headquartered here in Devon, Massachusetts, and we specialize in what we call emerging modalities, especially so all the complex chemistries and the new modalities such as peptides, so GLP-1 agonists is one of them. Oligonucleotide, they are short strand, mRNA. The name became famous a few years ago for good reasons. ADCs are antibody-drug conjugates. They are actually chemotherapy that we vectorize using protein so that it targets the cancerous cells and ignore the healthy cells. And the PROTACs, they are the protein degraders. So new super powerful drugs that are highly in demand right now with all the disease that we face. So my background, before I go there, I'm going to try to make a story out of the patchwork of experiences that you listed so people can kind of make sense out of that. So I have an extensive background in digital and laboratory technologies, like I say, beyond the test tube, beyond the round bottom flask. And so my background is in both biotech and pharma. I had leadership roles at Boehringer Ingelheim. We call that BI. Because it's a mouthful, and technology partners, companies making robots and automators and analytical system to understand what's going on in the test tube. And so Metla Toledo, we often call them MT, like BI, MT, you have a soup of acronyms to make it easy in people. And so my passion is driving, pharmaceutical innovation in chemistry and engineering. My background is chemical engineering. Using advanced tools such as machine learning, automation, robots to some level. Robots, it's not the Optimus, of course, but it's trying to get there slowly. And data analytics, so it's a combination of science and also data engineering. And the difference between the two is data engineering is trying to organize the data so that you can run models and get some data science on the data after it's being organized by the data engineering tools. And the ultimate goal is to get drugs to patients faster and to make that in a sustainable fashion. Try to pollute as little as possible. Pharmaceutical is a very wasteful industry, and we try to minimize the environmental footprint.
Okay.
And so fast, cheap, and environmentally friendly.
Good. So that takes care of the question of journey. So thank you for doing that. Let me ask you a couple of things. So first, how has your approach to innovation evolved as you've taken on more leadership roles? And then two, are there any challenges or opportunities in process development that you find especially energizing right now?
Yeah, no, absolutely. If life science and drug development was easy, we would know that, right? So we know it's challenging, we know it's costly, we know it takes forever to get a drug to the market. So my background, I guess, is unique in the way that I have navigated different worlds all my life. I grew up, got my education in Europe, came to the Boston area at the very beginning of the biotech revolution, I would say, at the end of the 1990s. And so I've seen all the evolution of biotech in the Boston area, but I also lived in other states, in other countries. Of course, I got my first job in England, but the company was headquartered in Cambridge, Massachusetts. And so I transitioned back 28 years ago now. Came to this world to be a true scientist, a true engineer, getting, in the lab. And then by force or by happenstance, I moved to the business side, so the dark side, as the scientists call it. And so I discovered business a little bit by force, but I ended up enjoying it because it gave me a different perspective of what it takes to get a drug to the market. When you're in the lab, it's difficult to imagine what's really at stake, what are the business drivers. And when I went to the business side, because I was selling multi-million-dollar ticket robotics, I had to talk to the C-suite level. They will be the one approving. And so through those conversations and lunch conversation and dinner, I discovered that it takes a very long time to get to the market, 10, 15 years sometimes. It takes billions of dollars, anywhere between $1, 2, maybe $3 billion to get a drug to the market. And that speed matters more than cost. I remember being stunned by hearing that spending $5 million was not an issue if they could get a bit faster, because it's $100 million a day sometimes in development. And so having navigated from being a scientist to being a business guy and back to being a scientist, I have a much better appreciation of what it takes in the lab to make a difference eventually, right? So I'm a lot more focused. I even created a company about 15 years ago to consult and bring my expertise to people beyond my everyday activity. And so, yeah, I guess it's having that multi-universe experience across countries and across specialties that gave me a different appreciation of what it takes to get a drug to the market and what the challenges are.
That's a good point. And, really, it's more of a symbiotic process, or a symbiotic relationship. Developing the drugs and getting the drugs to the market, it takes a lot of time. Didn't even talk about clinical trials, how many you have to have. It's really good to have that all-round experience from both sides of the spectrum. So which is
different phases in the development.
Exactly. Yeah. Let's talk about the science behind biotech companies in general. Okay, what are some of the most exciting scientific advancements you're seeing in biotech today?
Yeah, so if you look at a little bit of, open the newspaper, I would say, and you see what's going on out there. You hear about, there are different fronts actually in different, depending on the drug, depending on the therapeutic area. But back to COVID, let's say we got away of a technology that we call the mRNA technology, right? So it's making short strands of RNA to treat patients. So we developed the mRNA technology with a very broad scope of therapeutic disease, but we focused that for the past 5 years on vaccine against viruses. And that was good. It was welcome. But actually, you can go long beyond that. And today you have the same company, Moderna and Biotech and Pfizer, but we didn't get a chance. So the mRNA technology is one, it's extremely engineering intensive. It's extremely difficult to develop to to formulate and to deliver to the patient. Remember the minus 90 degrees reach that we had to have at the time of COVID to be able to stabilize and store the vaccine. So we made some progress. It doesn't take that much, but it's difficult still to formulate and deliver to the patient. Now, another area that you can read about easily is the Eli Lilly Novo Nordisk battle around the diabetic drugs, the GLP-1 agonist. And so again, broader application area, even though at the beginning they were only focused on diabetes, but it goes well beyond that. Now today we are treating obesity and probably in the future some other addiction and other problems, health problems. So I mentioned mRNA, GLP-1. Another category is oncology, very high in demand with an aging population in the world. So we used to have chemotherapy as the miracle drug, if you will, but it got the reputation back in the 60s and 70s that potentially chemotherapy could kill a patient before it kills the disease, right? And so today we have better tools. We actually have even more toxic molecules that we bind to a protein receptor, and that protein directs the drug, the highly toxic drug, we call that the cytotoxic drugs, to the tumorous cell and ignoring the healthy one. So they are antibody drug conjugates because it's a conjugate between a drug that's a cytotoxic to a protein or an antibody. To make those drugs, you still have to manipulate very toxic compounds, and we do on the site where I am right now. And so it's super complex engineering where you have to build containment in order to protect the operators from the toxicity of the drug. So different level of complexity compared to the mRNA, but extremely complex from a containment standpoint, because you cannot afford to have an nanogram of compound going out in the air and poisoning people. So yeah, very complex drugs. So GLP-1 agonist, mRNA, ADCs, antibody drug conjugates are very high up at the list of modern challenges in life science.
Very good. I read a book, I'm sure you've read it. It's called The Code Breakers. Mm-hmm. It was written by Walter Isaacson.
Yeah.
And it talks a lot about CRISPR and just the human gene editing. It's fascinating.
Yeah.
So it's a lot of heavy book to read, but it's really worth reading. How do these innovations, obviously gene editing is another thing that's a human innovation of time, but how do these innovations influence the way companies approach drug development in manufacturing? Can you share a breakthrough that changed our product? Was scaled or delivered?
Yeah, no, totally. I'm glad actually you mentioned the CRISPR-Cas9 technology, because we are really going now into a very different generation of drugs. So far, we've been talking about the kind of drugs that treat the cause but not the disease, right? So you basically apply a Band-Aid, hoping that it will get better in the future. But like the GLP-1, you have the debate whether you have to take them for life or whether after a while you can actually get off and go on with your life. And it's probably the former that's true, right? See, you probably have to take those drugs for life. So that's the traditional generation of drugs. So even though we have better technologies, they are still the kind of drugs that treat the cause but not the problem. And so with gene editing and cell therapy, we are going into a new generation where you take it a couple of times or maybe only once in your life, and then you're done. And voila, you're done for life, right? And so that's the dream drug. The problem is that, of course, they are not as well known, not as well mastered and controlled, and they have potential disastrous consequences. So it's never easy and a done deal that you can develop an entirely new system of operation and hope that it will work the first time forever. No, it doesn't work like that. So we had some cases where patients in clinical trials died from those therapy. So both of them are a miracle, but there's still years to go. And so some breakthrough over the past, you were asking, over the past 20 or 30 years, I guess you have. The first one that comes up to my mind is the recombinant DNA technology for insulin production, right? So it used to be that we would extract insulin from pork pancreas, and of course it's a dirty process. and very wasteful also. And so at the time we invented the recombinant DNA, we were able to produce insulin in vitro. And so that was breakthrough. But getting closer to the Northeast, and especially the Boston area, so I came at the very beginning of the MIT Novartis collaboration back in the early 2000s on continuous manufacturing. So to explain what continuous manufacturing is, when I go to my wife's school and give lectures on what life science and medicines are about, I usually take the analogy of the bath and the shower. Like, you have two ways of getting clean in the morning. One is to take a bath. It takes a long time. It's very wasteful. Or you can take a shower. It's actually a continuous process. It's faster and it's more sustainable. And so you can take the same concept and take it to a drug manufacturing. It's very childish, but that's a good way of looking at it.
That's right.
And so taking that concept to the world of pharmaceutical, back in the early 2000s, MIT and Novartis signed an agreement for something that at the time was about $60 or $70 million to develop continuous manufacturing and apply that to drugs. And you have a spectacular example back 10 years ago, published in the media, where Biotech Pharmaceutical developed a blockbuster against cystic fibrosis called Orkambi. And they were able, through that clean, efficient, space-efficient process, to actually manufacture the drug downtown Boston by the seaport area. And at the time, it was like, wow, for the first time, we don't have to put manufacturing processes, chemistry out of the wild, right? We can have it downtown. It's safe. It's clean. It's space efficient. And that was a very nice outcome from that MIT Novartis collaboration. And the last one, in the interest of time, was Merck. They have one of their blockbusters called islatravir, and they produce the drug by a cascade of enzymatically driven chemical reactions. So what are enzymes? In chemistry, we use catalysts to promote chemical reaction faster at a lower temperature in a more sustainable way. You can actually use compounds that they derived from cells, so that they are partially natural. They are clean organic enzymes that we use to make chemical reaction fast and cleaner. And that's how Merck got the Green Chemistry Awards about 10 years ago by being able to produce a drug by using enzyme instead of chemical catalyst and poisonous metals.
Let's talk about drug development. Now, obviously, there's, it's quote-unquote 5 stages, right? Like you have the discovery, development, you have preclinical research, you have the clinical research, you have FDA review, and then your safety and monitoring. That's just the core, but there's other things that goes into it. So from your experience, what are the biggest challenges in scaling the drug from lab to market?
Yeah, so there are many aspects, actually, but there is a whole area of scientific development in healthcare that's called translational research. And so it's a very enigmatic adjective, translational. It actually means, in simple term, in layman terms, the bench to the bedside, right? So we have many compounds that we discover that never make it to the market because eventually they don't translate from the test to the patient because they are toxic, because they're not scalable, because we cannot formulate them. And so that's the scientific and technical hurdle that we face when we go from the test to the bedside in the market. Another aspect is regulatory and compliance barriers. Of course, we want to have safe protection, so the drug that make it to the market and to the pharmacy are safe and efficacious. But of course, that slows down the process. And for a big pharma company, and most of the innovation come from, it can be the end of the journey because those companies usually operate on funds that they raise on venture capital and have a runway. So if the runway is 2 years and they don't make it to the market and don't make revenue by then, they may just end the journey and collapse and die. And eventually that's many potential drugs that would be fantastic but never make it to the market just because the regulation go against the financial model that we have today to finance life science innovation. And I would say the last one is just financial. Right now, we are seeing, back in September, there was on the first page of the Boston Globe, the biotech winter, right? So fundraising is more difficult right now after probably a period of irrational exuberance a few years ago, to quote Alan Greenspan. And so we are back to normal in the way that it's more difficult to fundraise, but developing innovation is extremely expensive.
Yeah, that's true. And you'll see it with some of our clients, especially getting funding just to get them, across or to extend their runway. We cannot have this conversation without talking about AI, right? In every aspect of what we do, AI is an important tool. So how do AI, automation, or data analytics play a role in process optimization?
There are different aspects. So you mentioned the different phases in clinical development. You have the pre-discovery phase, which is like the hit to lead discovery, right? Very early on, you discover some compounds that could be promising, and then you translate that to the test tube, and then to the animal, and then to phase 1, phase 2, phase 3, and sometimes phase 4, and then to the market. And AI can actually, today, I see the big gap in silico modeling for selecting the candidates that the most promising for bringing them to clinical. The attrition rate in pharma is gigantic. I was speaking with the head of a aerospace company last week, and he was saying that they could fundraise just fine. And it was like, why is it that VC would fund an aerospace company and it's so hard to get in money to biotech? Like for me, biotech is probably more important than I was, even though I work in that. And the guys say, I can explain that to you very easily, actually. In biotech, the chance of the upside is probably 1 to 100. And the chance of success is probably less than 5%. In aerospace, the upside can be 1 to 10,000. And the chance of success is about the same or higher. So I say that's easy. And so basically, if we could develop modeling using AI, machine learning, so that we select the candidates that we have a higher likelihood of making it to the bedside, that would be fantastic. So I see the big gap there. Like, how do we increase the chance of success going from preclinical phase 2B, that we call pivotal stage in clinical development, where actually the chance of success, there is an inflection point where it's very likely that the drug is going to make it to market. So like, I don't know the numbers exactly, don't quote me, but I think it's about 50% in phase 2B, which is like pretty high, right? 50% is pretty good. Things happen, but you almost make it there. And so, but in preclinical, it's probably less than 1%. So if we could go from 1% to 20%, then you can see that the upside is gigantic. That would be where AI has mostly. And then later on in clinical, I would see a big gap in robotics and automation especially. I was mentioning we make super potent drugs that are very difficult to contain and we don't want to poison anybody. If we could use robots to do that, that would be really fantastic.
So the probability of a higher preclinical trial to be successful has a almost same effect on a phase 2 success rate, right?
Yeah.
So to speak. What one tool or method you think is underutilized has huge potential?
Yeah, I would say so. Right now, I can see organoids. So I didn't even know what it was actually until recently, and I had to look up on Google like what, and that was related to some experiments we do on the ISS. So going back to a little bit of my background, I took 2 years rotation actually to go work in an aerospace company on the West Coast, and the idea is to try to leverage microgravity and the very harsh condition of space to bring innovation in all aspects. So it can be optical, physics, thermodynamics, but also life science. So we do conduct life science experiments on the ISS. And free flyers. So free flyers are actually independent spacecraft that we bring up to space because the ISS has limited capacity. And so I was hearing that Israel, actually, healthcare center, They develop organoids that they put in microfluidic devices and then they bring to their assay. So, of course, weight is a problem. And having full-size organs, even though you can 3D print in space, it's still challenging. So instead of using full-size organ to conduct clinical research, we print organoids. They are tiny, tiny organs that can emulate the behavior of a full-size organ. And so that's called miniaturization. So I believe very much that underutilized technology today, especially on Earth, is to miniaturize everything we do and try to leverage the power of the small to develop drugs in a more sustainable and faster and more economical way. And you have some example of that because it's related to what I mentioned before, that's continuous manufacturing. You have Eli Lilly, for instance, leveraging continuous manufacturing at one of its manufacturing sites in Ireland. And they can make more drugs faster and cheaper on a smaller footprint by leveraging continuous manufacturing. So it's not directly related to making things smaller, but making them more space efficient, which eventually goes back to the same problem. How can you be cheaper and more footprint effective by making everything smaller?
Yeah. Very impressive. You really just actually talked about process intensification, right? In terms of miniaturization, continuous manufacturing, end-to-end manufacturing. Are there any green chemistry initiatives or efficiency wins you are particularly proud of?
Yeah, so I was somewhat involved, not personally, but very close to my heart actually. Like, at the time of the MIT Novartis collaboration that I mentioned before, at the time the collaboration ended because the job was completed back around 2013, 2014, a few spin-off companies were born out of that. And one of them is called Continuous Pharmaceutical. And it's a play with words because continuous spells with that two-letter U's at the end instead of U. So you forget the O, and that's continuous manufacturing. And it was actually a company created to develop integrated drug substance, drug product manufacturing. So typically in a pharmaceutical world, you make the drug substance, which is the molecule, and then you pack it, you dry, you filter, you pack it, and you ship it to another site. And you make the drug product. That's where you mix the drug substance with the excipients. And you make the formulation, which can be a tablet or a capsule gel, or it can be an injectable, or it can be subcutaneous, depending on the application. And so the whole idea of continuous pharmaceutical is to say, why do we have two manufacturing plants? Why do we have two businesses, two partners, different location? Why don't we make everything on one site? And so the idea flew to some level, but then it goes back to the companies in the US. It's not the most affordable place to make drugs. And at the time, the business drivers were cost more than location. That's changing a little bit, right? Like location seems to matter more, and we have pharmaceutical companies investing in the US to make drugs in the US. But at the time, that was not the case. So the concept was great, but eventually it comes down to the business driver. It's definitely an intensification process because you make on one side, on a small footprint, everything. You don't have to ship material, sometimes toxic, from one part of the world to the other side of the world. You make everything in one location. It's a great idea, which may come back depending on what the business drivers will be in the next 5 or 10 years. And that's probably the example that I'm the most proud of.
Very good. So you just talked about things outside the laboratory, like the science. You talk about taking the drug to market. You have a couple awards that you've earned over time. The Green Chemistry Award and the Green Chemistry Award Principle adopted by CDMOs. Could you speak to that?
Yeah, so there is the Green Chemistry Institute, and right now I'm leading an initiative inside Veronova to take green chemistry to every single chemist. So we develop a tool that's Excel-based where we can assess the sustainability of a chemical process to a drug. And so I did something similar when I was at BI. When I was at BI, I was a data science And of course, evaluating the sustainability of process deals with a ton of data. So I use my data science expertise at BI to develop a tool which, in a way, I do a little bit the same here now. And so when I was at BI, actually, we got some of the green chemistry awards alongside Merck and GSK and Pfizer and Amgen, where those pharma companies are leading the way to make their processes use benign solvents. So benign solvents and probably no metal catalyst, but instead using enzyme. And so different companies, different strategy, but the end goal is to make those drugs use less natural resources, even using renewable resources sometimes. And of course, when those companies typically don't manufacture their drug, they do to some level, but limited, they rely on partners like CDMOs like us. And so when they take 5 years to develop a sustainable process, of course, they don't want their partners to use a polluting process. So CDMOs have to get on the bandwagon and get the same initiatives, take the same approach of using benign solvents and enzyme and lower temperature and reduced water waste and so on and so forth. So absolutely, I'm very proud actually of, after having conducted myself some green chemistry at Boehringer Ingelheim, to take that to the CDMO world in order to delight our clients.
Well, congratulations. So that is all the questions that I have for my friend Dom. We've touched on his career journey, which is quite exceptional. You know, we talked about the science behind biotech innovation and technologies and how they shape pharmaceutical manufacturing. Anything you'd like to add, Dom?
Yeah, no, thank you very much. It was a privilege to be invited to share some of my passion. The biotech landscape is changing very fast. Sure. So everything we said today would have been irrelevant 10 years ago and will probably be relevant in 10 years. So let's catch up again.
Absolutely, absolutely. So I wanted to thank Dom again for spending some time with me. I am your host, Cuddy, and we provide back office accounting support for levels ranging from staff accountant to CFOs. We support the offices of controllers and CFOs. And if you'd like to know more about what we do and how we support our clients, please go to witham.com/oasis. And that's spelled OASYS. Thanks again, Dom.
Thanks for joining us. Be sure to subscribe to our podcast so you'll be first in line to hear what's coming next. Don't want to wait for our next episode? Check us out at witham.com. That's WITHUM.com.